Hi everyone. Today in this video I'll explain some important bits which are helpful for the DSC TRT exam, that is 8th class biology. Now I will give some important points from the second lesson, that is cell is the basic unit of life. So I think this video will be useful for the people who are going to appear DSC TRT exam. So let's start. Cell, the basic unit of life. Introduction. Cell is a structural and basic unit of life. Some important scientists who are observing the living organisms which are not visible to the naked eye are Athanasius Kircher. He was born in the year 1601 and expired in the year 1618. John Swan Madam. He was born in the year 1637 and expired in the year 1618. Antonio van Leeuwenhoek. He was born in the year 1632 and expired in the year 1723. Robert Hooke. He was born in the year 1635 and expired in the year 1702. Anthony van Leeuwenhoek was the first human to see the living bodies like bacteria, yeast, protozoa and RBC in a drop of water. Discovery of the cell. In the year 1665, Robert Hooke, a British scientist, observed thin slices of cork from soft bark from the oak tree under simple magnifying device. He observed the cork as a honeycomb consisting of many empty spaces or empty boxes, and he thought cork was made up of very small cavities. And for these cavities, he has given the name as a cell. Cell is a Latin word meaning the little room. Observation of nucleus. Felica Fontana, she was born in the year 1730 and expired in the year 1805, was the first to see the nucleus in the epithelial cells. The meaning of epithelial cells, outermost layer of the animal body. So she observed in the year 18th century. Robert Brown, he was born in the year 1773 and expired in the year 1858, observed cells in the epidermis of orchid leaves. He observed it has a circular spot and opaque and integral part of the cell. He named it as a nucleus in the year 1831. There was a gap of 180 years between observation of cells and observation of nucleus. Observation of different types of cells. Observation of cells in the onion and cheek cells shows different types of a cell, that is cytoplasm, nucleus and cell membrane. Saffronin and methylene blue are two stains used in the observation of nucleus in onion and cheek cells. Cheek cell is considered as animal cell and onion cell is a plant cell. The boundary of a cheek cell is called a cell membrane. It gives shape to the cell. In the plant cell, there is another layer present over the cell membrane known as cell wall. It gives rigidity to the cell. In both cells, there is a circular spot called as nucleus. The jelly-like substance between the nucleus and the cell membrane is called cytoplasm. The jelly-like substance between the nucleus and the cell membrane is called cytoplasm. Organisms which have single cell in their body are called as unicellular organisms. Examples are amoeba, paramecium, chlamydomonas. Organisms which have more than one cell is called as multicellular organisms. Examples plants, animals and human beings. Amoeba has some projections on its body which appear and disappear. These are called as pseudopodia. In the word pseudopodia, pseudo means false and podia means feet. The smallest cell is found in the bacteria, which is 0.1 to 0.5 micrometers. A human liver and kidney cell is 20 to 30 micrometers in size. Human nerve cell is nearly about 90 to 100 centimeter. The largest cell is the egg of an ostrich. Microscope in which single lens are used are called as simple microscope. Microscope in which more than one lens are used are called as compound microscope. The first microscope was made by Zacharias Janssen in the year 1595. Usually we have 4 lenses on a microscope, that is 4x, 10x, 40x and 100x. Process of moving lens till image looks sharp is called as focusing. Measurements of a slide should be approximately 2 mm thick, 3 cm into 8 cm length and breadth. The material which we will observe under the microscope should be nearly 0.5 mm thick. The coloring agents which are used to the material to highlight a particular areas are known as stains. This process is called as staining. Examples: saffronin, methylene blue, Red ink iodine. To prepare saffron in solution, we have to dissolve 1 by 4th teaspoon of saffron in 100 ml of water. Here we have completed this lesson. If you find my video useful, then to continue making this type of videos, like this video and subscribe to my channel. Thank you.